Assalamualaikum. My name is Hafizi bin Nazli, S58998. My mini project is about early detection of highly invasive bivalve based on environmental DNA. Table content. It consists of introduction, problem statement, objective, methodology, result, discussion, conclusion, and reference. For introduction, non-indigenous invasive species or NIS can cause ecological, economic, and health harm to ecosystem. An early detection of invasive species should be conducted to avoid any circumstances that can be happen. The method that used to detect the invasive species is eDNA. It's defined as genetic material obtained directly from environmental samples such as soil, sediment, water, or matter. It used to detect DNA that is shed into the aquatic environment by cryptic or low-density species. Problem statement. How to detect invasive species that harm to the environment and ecosystem by using eDNA method. There is two objectives, which is to identify the best primer and also to identify the existing of invasive bivalve at the environment. For methodology, there is a few steps. First, primer design and laboratory sensitivity validation. First, design specific primer by downloading all 56 available mitochondrial CO1 sequence in the NCBI gene bank. Then took the high similarity of CO1 sequence of Limnoferna fortunei. Then the sequence were aligned in Mega software, and then designed the primer with fragment length ranging from 127 to 299 base pair using primer 5. Next step is field application. Sampling site at each location were randomly determined. At Pengji River, it is allotic water, therefore the sample were took from downstream to upstream to avoid contamination. Disturb the bottom layer before collecting water when that permitted is for lentic water. Next is DNA extraction. Extract the DNA from the sample and then quantify by nanodrop 2000 spectrophotometer. And the last step is PCR. PCR amplification was performed in a master cycle. Then visualize and analyze the PCR product using an automatic gelatin image analysis system. For result, based on the figure 2, primer B can be the best primer to detect the invasive bivalve. It is because primer B has high sensitivity and accuracy even though with the low abundance of the sample compared to primer 1. For figure 3, Pengji River has higher positive detection for both primer compared to other places. This happened because Pengji River has a huge population of invasive bivalve or cotton mussels compared to the other region and caused the high positive detection. For discussion, comparison between primer B and primer 1. Primer B are more sensitive to amplify the eDNA, while primer 1 is low sensitivity than primer B to amplify the eDNA. Primer B takes about 60, 40, and 20 minutes to detect bivalve, while primer 1 is take about around 90, and 60 minutes to detect the bivalve. The advantages of using high sensitive primer is can detect any non-indigenous invasive species or NIS that have long lag times before population exceed traditional detection thresholds. Next, the use of a sensitive primer pair on a field collected sample should reduce the possibility of a false negative result, which should be a key drawback of standard field sampling for rare species or species that just release only small amount of extracellular DNA. In conclusion, although the sensitivity of applied genetic markers and their related primer is rarely tested, eDNA-based techniques are increasingly used in the discovery of rare species. We observed that the sensitivity of different primer combinations varied greatly in this investigation, and we recommended that primer sensitivity be identified before field application. The use of extremely sensitive primer can reduce false negative results when combined with an optimized sampling strategy and solid field and laboratory procedures. This is the list of references that I use. Thank you.